Great. I think we'll get started now. Thank you all for joining us today at the Connect to Science seminar series. Before I introduce our speaker, I just want to give a couple of quick reminders to the participants. First of all, I'd just like to encourage you all to visit the website listed at the bottom of the title slide to get updates on the Connect to Science seminar series, including upcoming speakers. Also, during the presentation, if you have a question, please post it to the Q&A section at the bottom of the screen instead of typing it into the chat box. That way we can just organize the questions a little more effectively. So it's my great pleasure today to introduce our speaker, Dr. Yael Moshe. Dr. Moshe is Associate Professor of Pediatrics and the Patricia Brophy Endowed Chair in Neuroblastoma Research at the Children's Hospital of Philadelphia. Professor Moshe's impressive and prolific medical and research career began with her earning an MD from the Sackler School of Medicine in Israel. She then completed her internship, residency, and fellowship at the Children's Hospital of Philadelphia, where she also pursued postdoctoral research training in pediatric hematology oncology. She now runs a research group focusing on pediatric cancer genetics and especially neuroblastoma, with a particular interest in development of novel therapeutics. It is this interest that has drawn Dr. Moshe to help lead efforts in early phase clinical trials for novel agents in various pediatric cancers, and especially ALK inhibitors. Dr. Moshe has received an impressive array of awards and honors, including the James B. Nachman ASCO Junior Faculty Award in Pediatric Oncology and Career Development Awards from several foundations, including the American Cancer Society. We're so glad that Yael could join us today and look forward to hearing about our pioneering work in neuroblastoma, an area of major unmet clinical need. Yael, thank you so much for accepting our invitation. I'll now turn things over to you.
Thanks very much, Chris. Can you hear me? Okay, great. I appreciate the invitation very much, and thank you for that really kind introduction. It's a privilege for me to be here today and to tell you about the story of what we're doing in the lab. I'll start just with some disclosures. I'll only just mention that I am a paid consultant to Pfizer, and I'll be talking a lot about some of the Pfizer molecules, but my consulting does not impact any of the work that I'm going to present today. So when I started working in the laboratory after completing my first year of fellowship in pediatric hematology oncology, I had absolutely no really basic science training or laboratory background. My first formal research project was to find the familial neuroblastoma gene, and interestingly, it was at CHOP early on in the 1960s that Jane Chatton, one of our pathologists, reported a kindred who had multiple disorders, including neuroblastoma, in 4 of the siblings. And then, as we all know, it was Alfred Knudsen, along with Louis Strong, who then predicted that, like all cancers, there would be a predisposition to neuroblastoma, and that there would be a 2-hit hypothesis. Alfred even wrote that in hereditary cases, he would expect that about 20 to 25% of all neuroblastomas would have some type of a germline mutation. We had collected the largest collection of neuroblastoma pedigrees. This work started with Gerrit Brodeur back in the 1980s, and then was taken on by John Maris when he started working in the laboratory. And then this work was handed to me when I started in the lab as the families that we had been able to collect over the years. The disease does appear to segregate in some families, and when it does so, it is in an autosomal dominant fashion. But because of the lethality of the disease, this has not really been a rich set of pedigrees that one would have, for example, like in breast cancer or prostate cancer. But we did, in 2007, have advanced technologies, and we had collected some new families, and we were able to perform a genome-wide screen using a SNP array, 6,000 SNP arrays looking for linkage. And what we found was a single peak on the short arm of chromosome 2, really suggestive of a signal here with a LOD score of 4.23. And this is what led us to hone in on this particular region of the genome. And we were lucky because we had one family where you can see there's an affected grandmother who must have passed on the gene to her daughter, who's the silent carrier, but who went on to have 3 children with neuroblastoma. And we were very fortunate because this was the only pedigree that allowed us to, because of a proximal and a distal recombination here, allowed us to narrow the region of interest to one that had 104 genes. And back in the day, this was in 2007, we approached this one gene at a time, doing Sanger sequencing. My mentor at the time felt that MICN was in this region, and MICN, for those of you who are connoisseurs of neuroblastoma, know that it is highly amplified in about 40% of patients, discovered by Manfred Schwab back in the early 80s, and a predictor of a very inferior prognosis. And we also know that when there's forced overexpression of MICN using a tyrosine hydroxylase promoter in the mouse, they develop neuroblastoma. And so we thought MICN must be the neuroblastoma predisposition gene. And to make a long story short, we never found germline mutations in MICN. We went on to sequence a number of other genes in this region, and then landed on this gene ALK, which for me at the time, anaplastic lymphoma kinase, we knew already there had been a publication that showed that when you look at this SNP array of chromosome 2, there are occasional amplifications of ALK separate from amplifications of MICN. So they're not part of the same amplicon. And so this caught our eye, and it turned out that in fact, pedigrees such as this really taught us that the majority of families where neuroblastoma is inherited have germline mutations in the kinase domain of ALK. This is a unique pedigree because it's quite large. We have 3 generations. And for me, this really was critical to the discovery, because it was this young lady who first contacted me at the age of 16 who knew that we were studying the rare families where neuroblastoma is inherited. And she told me that she had a sister who died of neuroblastoma, and she had another sister who was being treated actively for the disease. And then she had a brother who, let me see if I can use my laser, maybe that might be better. Can you guys, I'll just use the mouse. So she had a brother who had a strange eye, is how she described it to me. Anyways, we went on to discover the point mutations in ALK, and it turned out that this young lady who was able to get me blood on all the samples that are annotated here also is a silent carrier of the disease. She went on later to have, to grow up and get married and have a baby, who was fortunate not to be a carrier of the mutant allele of ALK. And so these are the families that we originally published back in 2008. And the disease does segregate in an autosomal dominant fashion, as you would expect. And the most common mutation that we see in the germline is at this residue 1275. And it turns out this is also the most commonly mutated residue in tumor tissue and somatic tissues. And really it was a watershed moment for neuroblastoma because there were 4 papers that got co-published in Nature, and none of us knew that other groups had landed on ALK at the same time. And so this really was very exciting and provided me the opportunity as a clinician to really think about, what it might mean to be able to position myself at the interface of both the clinic and the science. And so I started to do a deep dive into what is this ALK gene. Interestingly, it was first cloned by Steve Morris when he was working in Tom Look's lab at St. Jude, and that was back in 1994. And it was cloned in a pediatric anaplastic large cell lymphoma cell line. We know that now 70+% of these rare inflammatory myofibroblastic tumors harbor ALK fusions, whereas in ALCL, it tends to be the same partner that ALK is translocated to. In IMTs, in fact, ALK is quite promiscuous and has multiple fusion partners. And we think that in fact impacts the the biology of this disease and the behavior of these tumors. But really it was in 2007, which was about a year before our discovery of point mutations in ALK, that a Japanese group reported translocations of ALK, usually with EML4 as the partner, in up to 5% of patients with non-small cell lung cancer, which it accounts for about 50,000 patients a year. And then we were the first to identify mutations in the kinase domain. So just to highlight the difference, that when there are translocations, we don't see the extracellular domain, but in neuroblastoma, the receptor is full length and remains intact, with the exception of point mutations in the kinase domain. And it's interesting because our work really has helped to inform what we now know to be mechanisms of resistance to ALK inhibition in diseases like non-small cell lung cancer, where they see acquisition of point mutations in the kinase domain. So we set out to really understand and catalog the ALK mutations in neuroblastoma. We were able to subject 1,600 tumors from patients at diagnosis that really spanned the full spectrum of this disease, all the way from the congenital form, the low-risk form, the form that spontaneously regresses, to the very high-risk disease. And we initially sequenced the entire gene, and still until we started to recognize that all of the mutations reside in the kinase domain. And there are 3 hotspot residues. As I mentioned, this one here at 1275 is the one that is the most common in the germline. It's also the most common in the somatic mutations that we find. And then these other 2 residues are the second and third most common. And then there are these other low frequency mutations that I'll talk a little bit about later. Regardless of how we looked at the data, whether it was the whole cohort or just patients with high-risk disease, the presence of an ALK alteration is prognostic of an inferior outcome. And we know that about 4% of patients, in fact, do harbor ALK amplification. This is mutually exclusive from a point mutation. So either there's a point mutation in the kinase domain, or the receptor is wild type, but there's high level amplification. And what we showed is that mutations are present in all risk groups, in all patients with this disease, even in patients with low risk disease. But really our attention is on the patients who face high risk disease, because these patients are subjected to such toxic therapy, and despite that have such inferior outcome. And I'll just highlight here that our original sequencing of ALK was using Sanger technologies. And so with that, we were able to at least predict that about 14% of patients with high-risk disease had an alteration in ALK. And when we first presented these data at an AACR meeting, somebody from Pfizer came up to me afterwards and said, you know, we have this drug and we thought it was a MET inhibitor, but it turns out it also inhibits ALK, and could you pour it on your cell lines? And so basically right after the AACR meeting, he sent me a vial of this drug that we referred to as PF1066 for quite a long time. And this is a panel of neuroblastoma cell lines here characterized for their ALK status. And you can see that on the y-axis is the % growth inhibition, and only cell lines that have an ALK alteration respond to 1066, but you can already start to see that there's a difference in response based on the underlying mutation. And then we honed in on the cell lines that have the mutations that we see in the patients. So in particular, this one that I mentioned originally, the 1275, that's most common in the germline and in tumor tissue. And when you look at its IC50 across a dose response curve here, you can see that it's quite sensitive to this drug 1066, as is the cell line we have with ALK amplification, whereas the cell line that has this mutation 1174 is less sensitive. And when we start to look at whether we can abrogate phospho-ALK signaling with this drug in the cell line that has the 1275 residue mutation, you see that we can abrogate the signal phospho-ALK with only 33 nanomolar of drug, whereas for a cell line harboring a mutation at residue 1174, we need much higher concentrations. It's really only at 333 nanomolar that we start to see responses. And this is exactly what we saw in our in vivo models as well. So when we treated mice with either vehicle or 1066, those that harbored the R1275Q mutation responded. I mean, we had never seen this in the lab. These are models that are developed largely from patients at time of multiple relapses and are, very resistant to most of our therapies. So this was really exciting for us, and it was an on-target effect that we could see where there was abrogation of phospho-ALK. But it started to become really clear to us that there was a difference in the response based on the underlying mutation. Again, you see here a model with a mutation at 1174. And while this difference is statistically significant, this is not meaningful for patients, at least to have a major impact. And again, when there is amplification, interestingly, it's also exquisitely responsive. So we really cataloged the ALK mutations from the diagnostic tissues that we obtained from the Children's Oncology Group. And what we did here is that we began to correlate the sequence variations with their oncogenic potency. And this started to reveal to us that, in fact, some of those low frequency mutations that I showed you in the beginning are unlikely to be oncogenic, whereas others have biochemical properties suggestive of intrinsic resistance to crizotinib. So you can see here on the x-axis all of the different mutations we identified from patient samples, and then here measuring their catalytic activity. And the colors annotate where these mutations lie within the kinase domain. R1275Q, which is the one in the germline and the most common one in tumor tissue, is of course an active mutation, but and it resides here at the juncture between the alpha C helix and the activation loop. And mutations that reside at F1245 and F1174, these reside in a different part of the protein in what we call the phenylalanine core here. And you can see that they're what we call almost hyperactivating. And it's interesting because we never see these in the germline. And so we think that these are likely not tolerated. That's why we don't see them in the germline, whereas all these ones here in blue, we do see in the germline. And you can see here the different transforming capabilities of the different mutations. And there are some that when they reside in the C lobe, they're just not predicted to be activating. And so this was really helpful in our early understanding of the differential kinase activation and differential sensitivity to direct kinase inhibition, that some of these, in fact, have primary resistance. What was very helpful for us early on is that the structure of the ALK tyrosine kinase domain was unexpectedly similar to the tyrosine kinase domain of EGFR. And this became very useful for us for understanding and predicting the consequences of ALK mutations. So for example, you can see that the R1275 side chain here contributes to the interaction between the activation loop helix and the alpha C helix, and it does so to stabilize the protein in an auto-inhibited conformation. And when the arginine is substituted for, by example, replaced by glutamine, this disrupts that interaction. But more importantly, this provided us an opportunity to gain more insights into this mutation here at residue 1174, which lies in a different region in the C terminus of the alpha C helix. And it contributes to this very kind of small, well-packed hydrophobic core. And when it's mutated, the size of that packing becomes actually reduced and weakens the auto-inhibitory interactions. But when you start to look over here, what we recognized is that the 1174 residue is also in direct contact with this motif, the DFG motif, which coordinates the transfer of magnesium ions that are involved in ATP binding. And when this residue is mutated, it removes the restraint on the position of the DFG motif, and this causes an increased ATP binding affinity, or if you think about it biochemically, a reduction in the Km for ATP. By contrast, when you look down here, where 1066 became the drug crizotinib, and what we recognize is that crizotinib makes no interaction with the DFG motif at all, so that altering the DFG motif by a mutation in residue 1174 should not affect ATP binding. Should not affect drug binding. And this really, these early observations for us provided a structural hypothesis for how a mutation at residue 1174, and it turned out to be the same for 1245, can increase the affinity of the ALK tyrosine kinase domain for ATP, but without affecting its affinity for ATP competitive inhibitors such as crizotinib. And this was really important because we were able to move crizotinib into a phase 1 trial. I put here in red the date of activation because it was almost exactly 1 year after all of the Nature publications on ALK in neuroblastoma that we were able to open this study of crizotinib in the Children's Oncology Group. And the reason also that I highlight here that the recommended phase 2 dose that we defined, which is 280 milligrams per meter squared per dose given twice a day, is equivalent to 200% of the adult maximum tolerated dose. And the reason that we really pushed the dose was because of our understanding of the competition with ATP for 2 of the 3 hotspot mutations. So the early understanding of the biochemical consequences of the mutations really informed the dose escalation of the clinical trial. Let me share with you some of the results of that trial. So we enrolled 26 patients with relapsed anaplastic large cell lymphoma. Sorry about that. And you recall that ALK was first cloned in ALCL, and although about the majority of these patients do very well with chemotherapy, about 25% of these patients do suffer a relapse. This is what happens when these patients get crizotinib. So this is a PET scan that shows disease all in the cervical neck, the anterior mediastinum, and down here in the spine. And after 1 course of drug, which is taken orally, as I mentioned, twice a day, you see absolutely no evidence of disease on this metabolic imaging. The overall response rate is 90%. And more importantly, the responses are not only robust, but they're sustained. And while it may seem, so these are all the 26 patients, that they're not necessarily sustained because the x-axis here represents the months of therapy. This is because these patients all had a complete response, and then the treating physicians took them off to take them to transplant because it is still felt, I think, in the lymphoma community that transplant is the ultimate curative therapy. exciting for us is that in May of 2018, these data led to an FDA breakthrough designation of crizotinib for patients, pediatric patients with relapsed anaplastic large cell lymphoma. But perhaps more importantly, just a few days ago, Pfizer received word that their supplemental new drug application to add pediatric ALCL to the label was approved by the FDA. And it was solely on these 26 patients and in a clinical trial that we designed in 2008, the very first targeted therapy clinical trial in the phase 1 consortium of the Children's Oncology Group. We also enrolled 14 patients with these rare inflammatory myofibroblastic tumors that harbor ALK fusions. And the data are similar to the patients with ALCL, where we also see robust and durable response rates and durable responses with an objective response rate of 86%. And you can see sort of the duration of therapy here on the swimmer's plot. It's really impressive. The cure for these kinds of tumors is only if there can be a complete resection with wide margins. They're chemotherapy and radiation resistant. And you can see this type of disease here that just melts away with crizotinib. And so we are now presenting the data to the FDA and requesting not only adding IMT to the label, but having crizotinib available as a upfront therapy, not just for patients who suffer a relapse, since there is really no known curative therapy in the setting of surgically unresectable disease. And then here is the cohort, the response in the patients with neuroblastoma who we know had ALK mutations. These data are not yet published. 20 patients I show you here with ALK mutations. The top two had ALK amplification. Contrary to our preclinical data, we saw no responses. I'll only say about that these are always patients who also have MYC-N amplification. ALK and MYC-N amplification, despite the fact that they're distinct amplicons separated by like 12 megabases. Patients with MYC-N amplification who suffer a relapse tend to be very refractory to any of our salvage therapies. In red are all the patients whose tumors harbored a mutation at the residue 1275, the one that I mentioned, that's the most common. And you can see some responses here up to about a year, objective responses in 3 patients. However, there are 7 patients with this mutation where there were really no objective responses. And then, as we would have predicted, the patients with mutations in residue 1174 had no responses. And we had no patients enroll with a mutation at the third hotspot, which is residue 1245. So a little bit disappointing, but in fact, really actually coincided, with our preclinical work and was not necessarily a surprise. But there are times when at least we see proof of concept that ALK, despite the fact that it's very difficult to turn off in tumor tissue, when it is present as a germline mutation, it really is a tumor initiating event. So we met this little girl, Edie, when she was just about 18 months. She was born with stage 4 neuroblastoma and found to have, at the time, bilateral adrenal tumors. And she actually had a third mass in her posterior mediastinum. So like multifocal disease. When we met her, she had already had 12 rounds of chemotherapy and 4 major surgeries. And it was just something about her, really, it was the presence of bilateral adrenal tumors that made me think that she had a genetic predisposition, despite having no family history of cancer, first child to young parents. And we consented the family to germline testing. And this was the very first time ever that we detected a germline ALK mutation in our clinically approved lab. And so this was very exciting to us, but we really weren't sure what that meant in terms of ALK inhibition. Would ALK inhibition be toxic when you have a mutation in every cell in your body? But it turns out that she had a complete response after 3 cycles of crizotinib. She stayed on the drug for 5 years, and she's now been off. Actually, I forgot to update this, but she's been off now 3 years. So that's pretty remarkable. Interestingly, when we tested the parents to see if this was de novo or passed on, we found that the mom was the carrier, at the time a silent carrier. But I'll just briefly mention that when mom and dad decided to have another child, and we did chorionic villi sampling, and were able to let the family know that the little boy would not be a carrier of an ALK mutation. However, mom developed neuroblastoma during pregnancy, and she presented just like her daughter with bilateral adrenal tumors. And we treated her as well with crizotinib for 3 years. And she's also been in a complete remission now for about 3 years. So we learned a lot of lessons early on in our experience with crizotinib. And the results from our trial underscored the importance of ALK across histologically diverse tumors. And that's been a lot of fun for me to be able to think about ALK beyond neuroblastoma. And the limited responses that we saw in neuroblastoma highlight the differences between therapeutic targeting of full-length ALK in neuroblastoma and of cytoplasmic ALK fusion proteins in diseases like anaplastic large cell lymphomas, the inflammatory myofibroblastic tumors, and non-small cell lung cancer. And little is known about neuroblastoma biology at the time of relapse, and yet, these are the patients that are exposed to new drugs. And so, what should the expectation really be when they develop multi-drug resistance? But at that time, crizotinib was really all we had. And crizotinib was, really it changed the field for non-small cell lung cancer for the patients with EML4 ALK fusions. And, I think we can only dream of seeing a waterfall plot like this in neuroblastoma. And the FDA granted accelerated approval of crizotinib for the treatment of ALK positive. And again, recall that it's ALK fusions in August of 2011. And crizotinib is the very first generation and very first ALK inhibitor ever tested. And so we thought to ourselves that, we wanted to move crizotinib forward. We finally had a targeted therapy for a subset of patients with ALK mutations in neuroblastoma. And so we piloted in the laboratory what it would look like to combine crizotinib with chemotherapy. And while this looks like a busy slide, it's just 3 models, 3 patient-derived xenograft models. Actually, A and C are PDXs and B is a conventional xenograft. And they harbor the 3 hotspot mutations we see in patients. And we randomized the mice to 4 different arms. Control in black, in red is crizotinib, in purple is chemotherapy, just like we give it to our patients, topotecan and cyclophosphamide for 5 days every 21 days. And then in green is the combination of crizotinib given with chemotherapy. So crizotinib is given every day, and the chemotherapy, as I mentioned, just like we do it in the clinic. And I'll really just highlight the outcomes that we saw in the models that harbor mutations that we know are intrinsically resistant to crizotinib. And you see that by the red line. But when you combine them with chemotherapy, when you combine crizotinib with chemotherapy, we saw just these really impressive responses. And we think this is in part due to pretty impressive induction of p53 and apoptosis. I mean, you can see even, with the chemotherapy that eventually the tumors break through and become resistant. And these were durable despite, discontinuing treatment here at about 13 weeks, even at 38 weeks, there was no regrowth of the tumors. And so these data provided the rationale for the COG phase 3 trial ANBL 1531, which is for newly diagnosed patients with high-risk neuroblastoma. And this study was activated in the spring of 2018. And on this study, we performed when a patient underwent a biopsy, part of that tissue was subjected to Sanger sequencing of the ALK tyrosine kinase domain. And any patient who is found to have an activating ALK mutation, as we've defined it in the laboratory, so they're very clearly defined in the protocol. These patients are non-randomly assigned to Arm E, and patients on Arm E get crizotinib as part of the backbone of therapy for high-risk neuroblastoma, which for those of you who are familiar with this disease know that the therapy is really incredibly dose intensive. It consists of 5 to 6 cycles of moderate doses of chemotherapy, during which time we collect stem cells that we use later on for rescuing patients from myeloablative therapy. They also undergo surgery here. And then they have 2 transplants. So we've shown that myeloablative therapy does improve outcome, not for all patients, but for certainly a subset. And so now our standard is to do this 2 times in a row and then rescue patients. And then they go on to get their post-consolidation therapy with GD2 directed therapy. So dinutuximab, a monoclonal antibody against the dicyaluganglioside GD2 expressed on the surface of the majority of neuroblastoma cells. And then we came up with this idea of a continuation phase where after that they would continue to take crizotinib for another year. That really was the idea behind that came from the experience in PH positive, Philadelphia chromosome positive ALL patients who, really that whole field changed with the discovery of imatinib and its role in that disease. But as that trial got going and as patients were being identified, we really were committed to finding a better ALK inhibitor. And while the data that I show you here are comparing crizotinib to this, again, PF now we know it as 3922, this is now called lorlatinib. I show you these data, these are both Pfizer drugs. What I don't show you is that in between here are actually every other ALK inhibitor made by every other drug company that we also got in the laboratory. All of them looked identical to crizotinib with the exception of lorlatinib, which really stood out. You can see that it's got this ring structure. And when we looked at it in both in biochemical assays as well as cell-based assays, what we found is that when you compared the IC50s with lorlatinib compared to crizotinib, we saw much lower IC50s. And interestingly, we saw that it didn't differentiate between the mutations. So there wasn't that differential sensitivity anymore. And the same goes here for the cell-based assays. We took this drug into our patient-derived xenograft models. Here is one that has a crizotinib-resistant mutation at 1174, a crizotinib-resistant mutation at 1245, and here is a crizotinib-sensitive mutation. So first I'll highlight that in blue are the cohort of mice that were treated with crizotinib. So you see really no effect here. And then in red is lorlatinib. And this was astonishing for us to see not only the responses, but the sustained responses that we saw in these xenografts. We call this PDX Felix. It's after what patient we treated in Philadelphia who came from the UK after our discovery of ALK mutations. When we met him, all we had was crizotinib. and we knew crizotinib wouldn't work, so we actually never even treated him with crizotinib. But that at the time of death, his parents gifted us with the opportunity to draw a large volume of blood, and we were able to establish a PDX in his memory that his family named Felix. And you can see here the really robust response with single agent lorlatinib. We actually took his model and studied two different schedules: lorlatinib given every day versus lorlatinib given 3 weeks on, 1 week off. And you can see that the tumors break through and develop resistance if you don't continue to really expose and have I think you, just like with BRAF driven melanoma, you need near or complete inhibition of the oncogene, and that needs to be sustained. Interestingly, in the model that harbors a mutation that does respond to crizotinib, we had never carried this experiment out beyond 4 weeks. What you see here in blue with crizotinib is that at about 4 weeks, the tumors actually, despite continued therapy with crizotinib, they become resistant. But the lorlatinib sensitivity continues, and the dose of lorlatinib also is a tenfold lower. So we became very enthusiastic about this this molecule and did a little bit of work to try to understand why it is so much more of a potent ALK inhibitor than crizotinib. These are just the results of a chemical proteomics experiment that we did, where we treated patient-derived xenografts with either vehicle, crizotinib, or lorlatinib, and then subjected the samples to mass spec. And what we found, first of all, is that lorlatinib is a very potent ALK inhibitor, and it suppresses ALK much more potently than crizotinib does. And we sampled tumors from 2 different time points. I'll just have you look at this graph of ALK here. We sampled tumors after treating the mice for 2 days and for 6 days. And you see here in red with lorlatinib that at 2 days, you see almost complete abrogation of ALK activation, ALK kinase activation. That's what we're measuring in this assay. And it is sustained at 6 days, whereas with crizotinib, we see only a partial downregulation of ALK activation, and it's not sustained. Despite continued treatment, we start to see the rebound in the ALK activation. So this provided us some insights into why lorlatinib is a better drug. So it's a more potent ALK inhibitor. So that's, seems obvious, but it was kind of nice to be able to really see that. And then we have explored, perhaps, are there other reasons why, is it acting on other kinases? It's not known to activate, to work on kinases that are involved in the G2 transition. But what we were able to show is that lorlatinib also inhibits important kinases like PLK1, CDK1, MELK, that are all involved in G2M cell cycle transition. This came out of a gene set enrichment analysis here. And we were able to validate this in an experiment that I show you here with the immunoblotting. When you treat tumors with lorlatinib, you see complete abrogation of phospho-ALK, whereas we don't see that with crizotinib. But with lorlatinib, we also see, for example, abrogation of phospho-SHP2, a really interesting phosphatase, as well as a validation that we do also downregulate this really critical G2M PLK1 kinase, giving us some insights into the mechanism of action of lorlatinib. We combined it with chemotherapy to see if there was a superior role for the combination. So this is getting back to a model that has a crizotinib-resistant mutation, also has MYC amplification, and the mice are randomized to either vehicle. Lorlatinib is in red. the green is chemotherapy, so just as we did it in the prior experiments, and the blue is the combination of lorlatinib with chemotherapy. So it's interesting that these tumors with lorlatinib, as we continue treatment, they also break through at about 10 weeks. But with chemotherapy, we see a sustained response. And perhaps even more impressive is that this model, NB1, that has MYC amplification and ALK amplification. And remember, I showed you that with crizotinib on our clinical trial, we had 2 such patients and they just blew through crizotinib therapy. Here, the experiment is, these are the mice treated with placebo. In blue are the mice, the cohort treated with chemotherapy. And then the cohort treated with lorlatinib alone and lorlatinib with chemo, they're overlapping. So you actually can't see the 2 of the colors. But we discontinued therapy at about 18 weeks. And whether they were treated with just lorlatinib or lorlatinib with chemotherapy, we've not seen any tumors break through. So we're really excited about this drug. And we were able to open a phase 1 trial of lorlatinib in 2017. Just briefly, it's a drug that you take by mouth once a day. It's extremely well tolerated. Its adverse event profile is much different than crizotinib. We designed this as a fairly standard 3+3 design. And we are looking to see whether there's any neurocognitive toxicity. This drug does penetrate the blood-brain barrier quite substantially. And the adult trials did see some neurocognitive toxicities at much higher doses. I'll just point out here that we started at dose level 1, 80% of the adult MTD, and we are now at dose level 5, which is 200% of the adult recommended phase 2 dose. We have 2 cohorts on this trial. One is for kids and one is for adults, because we do see adults with neuroblastoma, and for whatever reason, we're seeing that they have a higher frequency of ALK mutations. This is the characteristics of the patients so far that have been enrolled. So there are patients who've had prior ALK inhibition therapy, and these patients are eligible for lorlatinib. The median age is either 6 in the younger cohort or 22 in the older cohort. You can see that we even have a 50-year-old who's been on this drug. We've been able to get patients whose tumors are representative of all the mutations that we know are really oncogenic, including some novel alterations and ALK amplification. And then a lot of these patients do have MYC amplification, and these patients, unfortunately, no matter what, don't fare too well. We've not seen any dose-limiting toxicities with the exception of grade 3 diarrhea in a patient who's lactose intolerant. And we forgot to let them know that, unfortunately, lorlatinib is made with lactose. But otherwise, it's been extremely well tolerated, and the range of courses has been anywhere from 1 to 24. So there are patients who've been on this therapy for a long time. We show with our PK data, so these are the PK data for patients enrolled at dose level 1, 2, 3, 4. I don't have dose level 5 yet. I'll just highlight that at dose level 4, we're already seeing concentrations of the dose of the drug at that are equivalent to twice the adult recommended phase 2 dose, which is 100 milligrams. So we're already up to the equivalent of the adult dose, 200 milligrams. So we think that these exposures are important. A unique part of this trial has been the opportunity to do serial liquid biopsies throughout therapy. And I'll mention some early data we have from that experience in just a little bit. I just want to show you what some of the responses look like. This is a boy who has a very large tumor that's entirely unresectable, as well as bony metastases. But this is a mass that's encasing the jugular vein and the carotid artery. And you can see that with MIBG imaging, so metabolic imaging, really, after course 4, it becomes much fainter. And after course 10, our nuclear medicine docs are calling this negative. Although the mass itself does not change in size. So we're starting to appreciate different responses to targeted therapy. And then I'll show you this response here in an adult patient who had previously received 2 years of crizotinib, 4 months of alectinib, another second generation ALK inhibitor, who had molecular profiling before of a new lesion before starting lorlatinib. And we identified, in addition to the known ALK R1275Q mutation, these other mutations. I just want to point out the TP53 because we know that these patients are usually multi-drug resistant. But she had a complete response to lorlatinib after 2 cycles, and this lasted for 2 years. So as I mentioned, we're doing serial ctDNA profiling, and we've not done this before prospectively in any pediatric cancer trial. We've done this in collaboration with Foundation Medicine using their platform. And so far, we have, of the 35 patients enrolled on the study, 26 have more than one time point at which we've collected blood and subjected it to sequencing to look for mechanisms of resistance. And I'll show you just what happens to this one patient who starts off with a mutation at 1174. And as their disease responds, the ability to detect the mutation, so the variant allele frequency comes down. And then unfortunately, as their disease becomes resistant, we see not only the rise in allele frequency of the original mutation, but we start to see the emergence of new mutations. And interestingly, we're seeing new mutations in the ALK kinase domain, providing us some insights into the mechanisms of resistance. So all of that work has led to, and for me, it's really just such a privilege to be able to sit at that interface of the clinical and the science, because the science has really informed the clinical trial and vice versa. So I talked to you about ANBL 1531, the study for newly diagnosed patients with high-risk neuroblastoma. And we're now in the midst of a big amendment based on, and sorry, I'll put all this up here, based on the data generated both in the lab and the early data for lorlatinib in the phase 1 trial. And so we are amending the trial to replace crizotinib with lorlatinib. And we have a new analytical plan for these patients. I won't go into these data in detail. The patients currently, once the amendment goes through, who are receiving crizotinib will transition to lorlatinib. And we think that given the absence of hematologic toxicity and no overlapping toxicities, we really anticipate that lorlatinib will be tolerated much better, and therefore there'll be fewer interruptions. The other change we're making is we're finally getting into the modern era in the Children's Oncology Group and moving from Sanger sequencing to next generation sequencing. So we designed in my laboratory a gene panel that has not only ALK, but has also some of the other genes that we know are enriched for in patients at time of relapse. So patients, genes in the RAS/MAP kinase pathway, PTPN11, which is also mutated in neuroblastoma, BRAF, P53, and ATRX will all be on this panel. So we're really excited about that. And we think that this will provide us an opportunity to detect more patients with ALK mutations, not just patients who have clonal mutations, but perhaps patients even with subclonal mutations to see if they'll benefit from ALK inhibition. We have some modified eligibility criteria where we'll be allowing patients who have an ALK mutation with a VAF as low as 5%. And we're no longer going to be calling out exactly what ALK mutation is activating, because we've learned over time in a lot of sequencing that there are new mutations that come up that can be activating, even if we don't necessarily have the preclinical data to show that. Patients who we do have a cutoff, less than 5% variant allele frequency or mutant allele frequency. We will be following these patients very closely, but we don't know that they would benefit from targeted therapy, so they will not be assigned to getting lorlatinib. And then we're still going to be using FISH to define amplification. We are excited about the fact that we are doing this not only in North America, but we're doing this in collaboration with our colleagues in Europe. And this is really the first time that the European group, the SciPen group, and the COG group are collaborating. And our goal is not only to conduct parallel trials, but at the end of the trials, we're going to merge the data, and these data are going to be used by Pfizer to support their pediatric investigation plan. And so in order to do this, we've worked very closely with our colleagues in Europe to make sure that we align the key eligibility criteria, the dosing regimen for lorlatinib and the dose modifications and the timing of response assessments. And we'll be doing serial ctDNA collection throughout using a new platform that we've developed. So this collaboration is going to be supported by a foundation, Solving Kids Cancer, and they've called it the Titan collaboration, which I think is a very clever acronym. And these are just the two different trials, the COG trial overlaid above the SciPen trial. And so we're excited about this, this collaboration, which involves a lot of us in the COG, the SciPen, and as I said, Pfizer has been a fantastic partner and is really committed to using the data if the signal is as strong as we expect it to be for a new drug application. And finally, I'll just end on this timeline. I won't have time to get into some of the other approaches that we're taking, but this is really when we started in 2008, the discovery and sort of all the different parts of the work that I've talked about today, some of the trials that I haven't talked about where we've tried to combine ALK inhibitors with other inhibitors, and where we are now in 2021, amending our current phase 3 trial to bring lorlatinib up front. With that, I will not go into the fact that ALK also is an immunotherapeutic target. That'll be for another time. And I'll really just thank the members in my lab and all my collaborators and funding and all the amazing kids who really, for me, are the fuel for what we do in the laboratory. And thank you for your time today.
Thank you, Yael, for that wonderful talk. So I would encourage the participants that were here to ask any questions that they may have via the Q&A section at the bottom of the page. I do see we have some questions, but I wanted to start off with a question mostly about the differences between crizotinib and lorlatinib. You went through the details showing that lorlatinib does hit other targets, especially those that are important, kinases that are important for the G2S transition. And yet the side effects, the toxicities of the therapies seem to be greatly reduced with the lorlatinib, which is a bit counterintuitive. And I'm guessing part of that is just the reduction in the dose that you have to give in order to get a potent ALK inhibition. But are there other things that are contributing to the reduction in toxicities that arise in patients with the lorlatinib over crizotinib?
Yeah, it's something, of course, that we've been very pleased about, because crizotinib is not well tolerated. It's a very weak ALK inhibitor, and it targets has a lot of other targets. It is interesting that lorlatinib does target the SRC family kinases quite potently, in addition to the kinases involved in the G2M transition, and yet has such a tolerable adverse event profile. And the reason for that, I really don't know. I don't know if it's because of its structure. It's this macrocyclic ring that binds really tightly to ALK. And for that reason, we think it's better tolerated, only dosed once a day, and the blood-brain barrier penetration is also quite impressive. So yeah, I don't know, Chris, why the side effect profile isn't perhaps, broader based on the different targets of lorlatinib, but it's something that we're really trying to elucidate, more so for understanding what other pathways we could co-target to prevent resistance.
Yeah, that sort of leads into a next question. Do you feel like its targeting of other kinases could be contributing to its strong efficacy? And are there efforts underway by Pfizer or others to then modify the structure in order to hit fewer or more selectively certain targets to determine what contribution those other targets may have to the therapeutic effects?
Yeah, that is something that we're really actively looking at right now. So lorlatinib, I do think is really potent and active in ALK mutant neuroblastoma, not only because it's such a potent ALK inhibitor, but also because it acts through other kinases. And I think that that's really critical. And we've shown that, and that's work that hopefully will be published soon. So I don't think that Pfizer is going to be modifying the structure of lorlatinib. However, you could imagine combinations, for example, of lorlatinib and a SHP2 inhibitor that would really make sense to perhaps prevent relapse is what we want to do. We don't want to necessarily just use this in the situation where patients suffer a relapse, but I think these are giving us insights into other nodes that are important in ALK signaling, perhaps, and how we can develop novel combinations to prevent recurrence. I mean, with the G2M kinases, there are so many, not so many, but there are a lot that are targetable. There are actually PLK1 inhibitors in the clinic. There's one at least. And there are aurora kinases that are also really important as part of the G2M transition that we could target.
Yeah, those inhibitors seem to be quite good. They haven't been very successful clinically, but they are definitely available for clinical exploration. Okay, we can turn to some of the questions from the audience. We have a question from Satoshi Yoda. Did you see differences between crizotinib and lorlatinib in the cell cycle regulation, even at the same suppression level of phospho-ALK?
Yes, absolutely. Very much so. If I understand the question correctly, I mean, crizotinib, you need much higher doses of crizotinib to abrogate phospho-ALK in models that have mutations that we've defined to be intrinsically resistant to crizotinib. Whereas with lorlatinib, we can, at very low doses, even just 10 nanomolar, we can abrogate phospho-ALK signaling.
Impressive. There's also a question from Sophia Stupinski. Do neuroblastoma patients have increased rates of ALK-related lung cancer, or the point mutation in the kinase domain does not influence the likelihood of ALK EML4 fusions?
Yeah, we never see fusions in neuroblastoma. It's interesting that, and in the families where there are germline mutations, we don't see an increased prevalence of non-small cell lung cancer or anaplastic large cell lymphoma, sort of the fusion-driven diseases. So, very different. And we think, in fact, that the full-length ALK with point mutations signals very differently than the soluble fusions. And we're working with mass spectrometry to try to elucidate exactly how the point mutations in neuroblastoma signal.
Neat. I know that we're running out of time. Maybe we have time for just one more question. So in terms of the differences between clinical responses to crizotinib in the other cancers that you highlighted and neuroblastomas, what do you think is accounting for the higher rates of resistance inherent to neuroblastoma? Is it just the degree of dependence on ALK signaling in the mutated patients versus these fusions? Are there other inherent differences in the cancers that make them naturally predisposed to being very, very sensitive to crizotinib or resistant?
It's such an important question, to help understand the consequences of our work. I think that for anaplastic large cell lymphoma, and the fact that that is purely driven by an ALK fusion that almost always has the exact same partner, it's always ALK with the nucleophosmin gene on chromosome 5. I think that that is a disease that has a pure oncogene addiction, and where we have not seen resistance in these patients. And so it's really quite a pure disease with no underlying biological heterogeneity, at least none that we've been able to elucidate. Whereas for non-small cell lung cancer, it gets a little bit more heterogeneous there because the patients do respond very nicely and then develop resistance. Neuroblastoma, I think, is just even more complex, the underlying biological heterogeneity, and especially at time of relapse, upon the selective pressure of our therapy, the genome really evolves and they develop mutations in the RAS/MAP kinase pathway, in other pathways, and while they still have a partial ALK addiction, they are also really impacted by MYC/N and RAS and ATRX. We think that the time to make a difference is at diagnosis, and that's why we've really pushed to amend ANBL 1531 for the completion of that study, knowing that we have a better drug. And certainly, my hope is that at diagnosis, when these patients are chemotherapy naive, we'll have a better chance to really get the ALK-addicted tumors to turn off and stay off.
Excellent. Yeah, that seems like the most rational plan for effective therapy. We do have to stop here because we're out of time. Dr. Mosay, thank you again for joining us today. It was a pleasure. Wonderful work that you've been able to do and share with us today. So we hope to see you again soon, and thank you again.
Thanks very much for the invitation, Chris.